Introduce our next speaker, Professor Hong Ma from Oregon. Will be indeed talking on mitochondrial diseases, preventing mitochondrial diseases with so-called three-parent IVF. Welcome.
First of all, thank you very much for inviting me to attend the conference. I came from Vitaly Popov lab at Oregon Health and Science University. Today, I will share some new studies we recently conducted. Cell therapy. Definition of cell therapy is very straightforward. It's administration of live cells or maturation of a specific cell population in a patient for the treatment of disease. Nowadays, IVF is a standard procedure for infertility treatment. We think IVF itself is cell therapy. In IVF, embryos are produced by fertilizing eggs and sperm in a dish and maturated through in vitro condition. IVF embryos can be used for treatment and also can be frozen for future implanting. We think IVF is very good platform for germline gene therapy. Germline gene therapy is correct or replace mutated genes in germ cells, and genetic correction will be passed on to the next generation. And currently, germline gene therapy is still in its infancy. There are many restrictions on research of human germline gene therapy. And not every state in the United States are allowed to do research on human germline gene therapy. We are very lucky in Oregon that regulatory environments allow us to do research in this area. So our interest is for germline gene therapy for the disease caused by mitochondria mutated gene. We all know mitochondria play a central role in the energy production. More than 90% of cellular ATP are generated from mitochondria through Oxford system. Mitochondria contain their own DNA, empty DNA. Sorry, I pushed it wrong. Can I get back?
Yeah.
And
you can go back. There's the arrow.
Okay, it's okay. And contains its own DNA, empty DNA, which is separated from nuclear DNA. Empty DNA inheritance is exclusively through female lineage, from mother passed to children. And each cell contains multiple copies of empty DNA. The number varies, depends on the bioenergy needs of each particular cells. Empty DNA encodes 13 core subunits of 4 protein complex in the electron transport chain. In addition, empty DNA encodes 24 RNAs, which are essential for protein synthesis of these 13 polypeptides. Like nuclear DNA, empty DNA is prone to mutation. The first disease-associated empty DNA mutation is reported in 1988. Since then, more than 700 disease-associated empty DNA mutation has been discovered. Some of the mutations are inherited, which means mutation arise in female germ cells. This type of mutation are cause of inherited mitochondrial disease. Most of mtDNA mutation are acquired, which means mutation arise sporadically in somatic cells and accumulate with age. These type of mutations are associated with late onset common human disease. Our interest is maternally inherited mitochondrial disease. Maternally inherited mitochondrial disease is caused by pathogenic mutations in mtDNA, which results in either fatal or severe syndrome in children. Each year in USA, up to 4,000 children are born with inherited mtDNA syndrome. Currently, there is no cure. And in some cases, PGD may help to select mutation-free embryos, but however, PGD does not always success due to the complex inheritance of mtDNA mutation. We propose a strategy, and it's called mitochondrial replacement therapy to prevent transmission of mtDNA mutation to the next generation using spindle transfer. The concept of MRT is we take a spindle from patient's oocytes and transfer it into a donor oocyte. Healthy oocytes came from donor, which with spindle being removed. This reconstructed oocytes will be fertilized to become healthy embryo. Eventually, a healthy baby can be born. In MRT, genetic materials come from 3 individuals. Nuclear genome comes from patient and sperm donor, and the mitochondrial genome comes from healthy egg donor. We initially test safety and feasibility of spindle transfer in non-human primates. In this case, we collect eggs from 2 different monkeys. One is India origin, second is Chinese origin. They have mtDNA sequence different. Using spindle transfer, we successfully generated 5 healthy newborns. First 4 are male, the last one, Krista, is female. Now most of them have reached reproductive age. And during spindle transfer, a small amount of maternal mtDNA carryover is common. It's usually very low, below 2%. To monitor carryover dynamics in vivo, we collect blood samples from 3 monkeys at a different time point: newborn, 1 year, and 5 years. This data show the carryover did not change with age in monkeys. We also paired 2 male monkeys, and they both made a female monkey pregnant. This year, 2 healthy female baby was born. We named them Nora and Mia. These results show that MRT does not affect reproductive function in the next generation. This is an ongoing study. After we obtain promising results from non-human primates, we translate MRT to human study. Initially, we performed MRT in healthy human embryos and generate embryonic stem cells. Those embryonic stem cells appear to be normal. And this year, we conducted a preclinical study using eggs from women who carry mtDNA mutation. As shown here, these two women came from a family with 8993 T2G mtDNA mutation. They both had a child with Lee syndrome. We collect eggs from these two women and performed spindle transfer. Replacement efficiency is very high in our hands. All ST embryos contain higher than 99% donor mtDNA. Because the different mtDNA sequence, or we call it haplotypes, present within human population, there is an issue of haplotype difference between patient and the donor. And to address if mismatched donor haplotypes affects embryo development, we divide ST embryos into 3 groups based on the distance between patient and the donor as measured by SNP difference. The close group is 6 SNPs, middle 33 SNP, the most distant 57 SNPs. And there is no difference in normal fertilization and blastocyst formation among 3 groups. And these results suggest pre-implantation embryo development was not affected by mismatched donor haplotypes. However, mismatched donor haplotypes may affect donor mtDNA stability. We observed 3 ES lines was gradually lose donor mtDNA and return to the maternal mtDNA haplotype. So we consider two mechanisms might able to explain this reversal phenomena. First is CSB2-dependent preferential replication. Sequence conserved block 2 region is a GC-rich region in the D-loop of human mtDNA. This region functions as a switch to stop RNA transcription and start DNA replication. Sequence difference in this region is associated with different replication efficiency. We found in 2 reversal stem cell lines, and there is a polymorphism in donor mtDNA H1b haplotype, a single C residue deletion. This polymorphism results in 4-fold reduction of donor mtDNA, which means maternal mtDNA replication much faster than donor mtDNA eventually takes over. Second mechanism is haplotype-associated cell growth advantage. And the remaining 1 ES cell line, there is no sequence difference in CSB2 region. In this case, we observed cells contain maternal haplotype H49 grows significantly faster than cells contain donor mtDNA. So this suggests some haplotypes may give cell with fast growth advantage. I have to point out, although this reversal phenomena was found in ES cell lines, and we believe it can take place in vivo too. Here comes MRT highlights. This is the first form of germline gene therapy. It's use of mitochondrial genome from healthy egg donor. So MRT is suitable for any type of mtDNA mutation, homoplasmic mutation or heteroplasmic mutation. our preclinical studies demonstrate safety and high replacement efficiency. We recommend donor mtDNA haplotype matching to prevent maternal mtDNA reversal phenomena. Now I'm going to switch my presentation to the second part. We know pregnancy rates decrease as women get older. At the same time, miscarriage rates increase. And this data show that live birth rates as a function of woman age in the IVF clinic. Interesting is when women use their own egg, the pregnancy or live birth rates decrease dramatically, particularly after age of 40. Same aged women, if they use donor eggs, which usually from young women, the live birth rates keep the same. So this tells us the reduced fertility potential in women with advanced maternal age is oocyte related. And clinical treatment options are very limited if they want to have their own child. So we developed a new procedure using IVF platform, which could potentially increase patients' related oocytes available for IVF. Mature M2 oocytes cytoplasm contain meiotic activity. This activity can reprogram donor nucleus if transferred into and to form the oocytes like spindle. Women are born with a quite number of eggs which are arrested at meiosis 1. After completion of meiosis 1, the first polar body is separated from oocytes after fertilization. Second polar body are extruded from oocytes. Polar bodies are discarded genetic materials. They do not participate in embryo development. So we think if we can recycle the first polar body and use it to create another oocyte which related to patient, this schematic show our new procedure. It's called the polar body nuclear transfer. If we take first polar body from patient oocytes and transfer it into a donor oocytes with spindle being removed, this reconstructed polar body oocytes can be fertilized to become an embryo. So from one single patient oocyte, we can potentially produce 2 embryos available for infertility treatment. So this data show that polar body embryos had similar normal fertilization, but reduced blastocyst formation compared to control group. This suggests nuclear contents in some of polar body embryos are compromised. So we think by optimizing our procedure or protocol, we can improve the blastocyst formation rates. Top panel showed aneuploidy rates. The bottom panel showed subchromosome level changes. Because aneuploidy is common in IVF embryos, which could result in implantation failure. So we performed array CGH in blastocysts in polar body group compared to control. And the bottom is for the subchromosome level changes as structural variation performed by genome-wide physical mapping analysis. Both data showing there is no difference between polar body nuclear transfer group compared to control. And lastly, we performed DNA methylation and transcription analysis in ES cells derived from polar body nuclear transfer embryos and from control IVF embryos. Left panel is CD methylation, middle is non-CG methylation, the right panel is RNA-seq data. This data showing global methylation profiles and gene expression pattern of PBNT ES cells appear very similar to control IVF ES cells. Here's a summary. Genetic materials of discarded first polar body can be rescued to generate functional haploid oocytes through with young donor cytoplasm. Polar body nuclear transfer technology of a novel source of patient-related oocytes available for IVF. Also, polar body oocytes provide another MRT approach for mtDNA-based disease. There are limitations. First, PBNT technology is not beneficial for women who cannot produce mature oocytes. And second, PBNT will be limited to countries where IVF programs are allowed to coordinate oocytes donation cycles. Lastly, polar body nuclear transfer technology will have to meet safety requirements of regulatory agency before approval for routine application. Lastly, acknowledgements. So all the human study are collaborative work between our lab and the OBGYN department led by Dr. Amado. And my special thanks to Dr. Belmonte and his team, to Dr. Aker and his team from Salk Institute, and also acknowledge Dr. Dmitry Temiakov and his team from Rome University. Lastly, acknowledge our funding agency. Without their support, we cannot complete our mission. Thank you very much for your attention.